as I've said, with genomic data analysis, you have a variety of flavors of assays that measure different molecules in your biological context. And with genomics, we can do all of the molecules together, all of the species together. Each genomic assay is going to have its own underlying statistics. So you need to make sure that the analysis tools you use are built for that kind of data. Why am I saying this? Yesterday, I was at a poster session and there was somebody doing epigenetics of nerve pain. And it was like, we're using scikit-learn and we're trying to like A versus B and t-test and p-values and no, right? So this course is a lot about trying to kind of think about your data and think about the confounds in your data and how you can ferret them out and clean your data out so that you understand it and you know your data before you start throwing it into pipelines, right? A lot of times it's like very pipeline centered. Like I got RNA-seq data, boom, I'm going to run this package, that package, whatever. But we are going to focus in this course more on trying to understand the concepts underlying some of these things and best practices. And I strongly urge you, unless you want nasty surprises halfway through your project or close to publication, to do this due diligence upfront, right? So we're going to start with exploratory data analysis. Okay. So don't get scared the moment I throw up the word statistical, right? We're going to work on this. But by the end of this lecture, you will know basic terminology for a statistical model, which is the heart of all of our sort of biomarker finding the cool molecules that do X, Y, Z, right? So we're going to understand the basic terminology for statistical model. You're going to learn, we're going to give you a formula for how to systematically perform what's called exploratory data analysis. This is the first step of any study, right? Before you start doing your A versus B, you have to look at your data. You have to think about what are the things that could be influencing the data and the way it looks. What do you need to correct for before you launch into your statistics? All of that is called exploratory data analysis. Please budget time to do this for every data set you have. So we're going to talk about how to perform data analysis, exploratory data analysis, or EDA, to identify sources of biological variation and technical variation. Then we're going to talk about exploring missingness in your data, right? So some assays, you're going to, not have any kind of missing data whatsoever. There are some high throughput genomic measurement technologies such as microarrays, like no missing data usually. But in the real world, with a lot of different data collection methods, you are going to have missing data. So you need to have a step in there to understand what is the nature of missing data and what can you do about it. When do you, extrapolate or interpolate to fill in the missing values? When do you throw out samples, etc.? Then you're going to have a high level understanding of clustering, which is a way of finding groups in your data, groups that may reflect biological entities or technical variation, right? But it's sort of the foundational thing when you're analyzing multiple data sets is how do you cluster? And, PCA is one method for clustering, but clustering is a general term to mean you take a bunch of data points and you assign them into groups, right? So PCA is one tool for it. So let's start, with something very basic. So suppose your goal is to find out, you're interested in a disease condition, say, right? And you have two groups of data, A versus B, control versus case, right? People who respond to medication, people who don't respond to medication, to keep it simple. And suppose your goal is that you are going to measure 20,000 genes in all these patients, and you want to find out what are the genes that are different between A and B, right? So find the transcriptomic biomarkers of disease. Okay. So at the heart of it, how do you translate the sort of sentence into math, which is what your algorithm or your software packages are going to do when you say differential expression, differential whatever, right? This is at the heart of it. What's going to happen? It's going to say the expression of a gene can be explained by some kind of background, offset, plus a certain amount that depends on disease, right? So in the plus unmodeled residual variation, things that you didn't consider in your model. And there could be a lot hiding under there. We're going to talk about it. But this is just to say the expression of a gene, is a function of some kind of offset, right? That may be due to your assay you're using, plus some kind of factor associated with your disease that is non-zero. The non-zero ones are the ones you want to find. If a gene has nothing to do with disease, this would be 0, and the gene's expression would be like the same throughout all your patients, right? And then you have this unmodeled variation. So in statistical terminology, this is called your response variable. This is your explanatory variable. Explanatory variable is something that explains the value of the measurement, right? And this is called your residual, your unmodeled variation, right? So if you strip away the Greek letters, it's basically just like, you can translate this. You're just saying, I think expression is either due to like some kind of baseline plus something to do with disease. Another way to think about this is that you have a dependent variable, which is expression, and then you have independent variables that determine the outcome of the dependent variable. This is just a terminology, right? Embrace the terminology. Betas are called the coefficients or the weights, okay? And this is the value that under the hood, your differential expression analysis, et cetera, is going to tell you, right? If the beta is non-zero, if beta 1 is non-zero, then that gene has some kind of statistically significant disease effect, right? And this is called the intercept. You always need some kind of offset value to account for, like, maybe everything starts at 5 and it's 5 plus or minus something. So you need a term to kind of put that into. Okay, so this is what we've got here if we want to find the transcriptomic biomarkers of disease, right? This is going to be true whatever your response variable is, proteomics or whatever, right? Now, are those the only things that could be affecting the expression of the gene in your data set? Or could there be other things? I want you to think about, like, if you're thinking about a population, right? And you want to find the genes associated with, I don't know, diabetes, right? And you collect data from people with diabetes and people without diabetes, and you want to find out what genes are associated with diabetes. What other sources of variation could be affecting your data? Okay. Broadly speaking, variation can be in 2 flavors. One is biological sources of variation that have to do with the underlying population you're studying, right? The second comes from the fact that we are measuring it using an imperfect technology in an imperfect lab environment, right? So it has to do with measurement variation. If everything was perfect, you got perfectly reproducible results every time, your technical sources of variation, you had the perfect assay, it had 0 bias, then that would be 0. And then you just have to worry about biological sources of variation. But this is just to say that when you look at your data set, you need to think about how are the data generated? If you are just getting data from somebody else, you need to get a table that tells you other associated information about how the data were generated. So I'm asking you guys now, what are some examples of biological sources of variation?
Sample size, if you're like working with a population of patients, like are you, if you have incomplete coverage of a population, for example, or it's not like representative of everyone in that, does that count as a source of biological variation?
That would be biological, but this would be, you would have to pinpoint what that biological variation could be due to. So what about it? Exactly, right? So sex is an example of biological source of variation. What are other biological source of variation. Just, this is not hard. This is straightforward, huh? Ethnicity, yes, it's huge, especially if you're doing genetic studies or even gene expression studies. Yep, family history. You can find a way to encode it in a table. You want to build that into your design. What else? What else are other source of variation? Mm-hmm. Mm-hmm. That would be sort of intersample variation, but in the wet labs, let's say you're in the lab, right? What are some technical sources of variation that might arise? Yeah. So how would you encode that in your model? Pipetting variation. Mm-hmm. Suppose you have a data set of 1,000 people that you ran genomics on, right? Now, were all 1,000 of those samples, was the data generated by the same person? Was it all generated on the same day? Was it all generated with the same reagents? Right? So what are examples of technical source of variation? I pretty much walked you to that.
You know, you can have write down the batches.
Batch effects, right?
So you have batch numbers, so you compare between batches of
Yeah, I can tell you that when I've looked at microarray data, which is a kind of high throughput measurement before people start doing sequencing, people still use it. The lot number of the microarray can make a difference. So if half of your, like a third of your samples are processed in lot A, and then a third on lot B and lot C, when you do exploratory data analysis, you will see lot A sitting by itself, right? Technician variability, right? The one time you had the summer student come in, or something happened, the person found out about a grant that didn't get funded, right? Like the Tuesday effect or the Wednesday effect or whatever. So the point is, you need to be aware when you look at your data that they are being affected by these things, batches. So like we generated a pilot data set, everything looked good, so we added more samples to it. The whole second time process, those data are going to sit by themselves. In fact, it's one of the biggest sources of variation. If so which is why EDA, visualizing your data, using tools like PCA are going to help you visualize it and quantify it and then account for it in your model so you can subtract that effect. Okay. But if you're not even looking for it, you just kind of throw in, you don't look at it and you call differences. What if your differences are due to Monday versus Tuesday? Nobody wants to go out and publish a paper. So yeah.
Kind of, from a statistics standpoint, it's basically confounding factors, identifying the confounding factors and trying to control for them.
Yeah, trying to build them into your model, right? So for example, if you're doing a human study, you better have age and sex in there as your controls. If you're doing a genetic study, you want to include at least 10 principal components from genetic ancestry into your model. Because I once saw this rebuttal on this autism genetic study where the original paper claimed that, this particular genomic loci, these SNPs, were associated with ASD cases versus none. They made some story like blah, 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 the biology. And then this group of geneticists, which is everybody's worst nightmare, wrote a rebuttal letter saying, this was like a Nordic study, like it was like in Northern Europe. And they said, look, we reanalyze your data. You've got slightly more Swedes in your controls compared to your cases. And that population stratification is what gave you your signal. So, EDA is the unsung hero, just makes me so paranoid. So, right? Sex, age, ethnicity, cell type, right? So if you're using bulk tissue, right, and you're doing like postmortem brain studies, and you took like you went to a biobank and you got like brain studies, or you, and then you got a bulk assay and you're doing RNA-seq. This is, single cell is nice in this way because everything's kind of laid out there. But if you're doing bulk, do all of your samples have the same proportion of neurons compared to the other cell types, Yeah, exactly, right? And there are techniques now to infer what your cellular mixture is. Like they will look at the neuronal gene and they look at a sample and say, oh, yeah, you've got 70% intensity of neuronal gene in this sample and 10% in that sample. So we think this is 70% neuron and that's 10% neuron. There are methods to do it, but you have to be aware of it and go through those moves in order to account for it. Otherwise, you're just kind of, your inferences could be faulty. Technical,
Sorry, on that, like we're, the, microbiome, class, we're talking about, metagenomics. Can you, with bulk samples for like RNA-seq stuff, can you use metagenomic techniques to separate what kind of cell types are present in the bulk sample?
Yeah, I imagine it's a similar process. So here's what you need to do. You need to find out the workflow. You need to find a review article that talks about the state of the art workflow for the assay of interest. And usually, there'll be some leaders in the field who kind of keep on top of the method development, and they will say, this is the pipeline. Or you take like a landmark study that the field believes is sort of like done, with sort of gold standard, and you look at their methods and what controls they're using, right? And they are unique to the data set. So for example, for DNA methylation, if you're doing bisulfite conversion based methylation estimation, bisulfite non-conversion is a QC metric you have to have. You don't have to do that for RNA because it's not part of that workflow. So some things are going to be, you need to think about where your data coming from, how are they generated? And when I worked with a wet lab that was really good at generating omic data and throwing it at the informatics people, we always asked for a spreadsheet with information like batch lot number, the parameters that are usually the culprit. And sometimes you just write R scripts that are going to do things like clustering and PCA and color code your samples based on variable 1, variable 2, variable 3, variable 4. Then you just go get a cup of coffee and you sit down, you just kind of like look at them. And then you're like, oh, there's a lot effect, right? So sometimes you discover these things, but you have to systematically go. Sometimes the field knows because people have done it enough. But yeah, I can't tell you, I've found duplicate samples, like males listed as females, neurons listed as non-neurons, and EDA will catch all that. You clean it up, then you can be more confident about what you're seeing. Exploratory data analysis. Yeah. Yeah, so the reagents, what was the antibody used here versus there? Everybody knows, like, once you find an antibody, you kind of hold it close to your heart. You buy up all the batches because the moment the company makes another batch, who knows? We once were training, I did animal neuroscience in grad school, and we were training rats to do this decision-making task. And suddenly all my rats started performing badly. They just weren't learning. And it was like weeks, and we were like, what happened? And then it turned out that our facility switched the provider. They moved to a different company from which they were getting the rats. And we don't know how these poor rats were raised, but they weren't learning nothing. So then we went back and we switched back to like provider A, and suddenly things went back to the way they were, for better or worse. And the other thing is, you might not know, right? It's like you don't know what you don't know kind of thing. So there are these visualization tools will help you identify structure in your data, and then you have to understand where that structure, what is contributing to the structure. Is it batch? Is it whatever, right? So which of these is affecting my data, right? Maybe not all of them for all of the data, but you have to know what is affecting your data. And it's definitely sort of a meditative thing. You look at your workflow for whatever your data set are, and then you find out what the best practices are and what the pitfalls are for your particular assay. And then you develop a toolkit using some of the techniques we teach you here, and you make your little pipeline, and then you just sit and look through it. Okay. So at the end of the day, you use methods such as clustering dimensionality reduction, which sounds really sci-fi, but it's very straightforward. We'll get into that later. These things, acronyms you've heard, such as PCA, UMAP, and TSNE for the single cell people. And at the end of the day, your statistical model becomes a little broader, right? It includes these sources of variation. You say, now I believe that expression of a gene is the offset plus the factor I'm interested in, plus these biological sources of variation, plus these technical sources of variation, plus the unmodeled source, right? The epsilon. So that is the goal of this exercise. Okay. Now, just a minor point about the epsilon, right? Usually in statistics, they say, oh yeah, the unmodeled variation is random. But when they say random, they don't mean arbitrary. They mean randomly sampled from a particular statistical distribution, right? In this case, it's a normal distribution usually. So any questions at this point? Okay, good. Explanatory variables, it's increased. It's gone from disease to all the others. These are the biological variables. These are the technical variables. And that's your random variation. Okay. What are sources of missingness? Let's go. If I keep talking, you guys are going to fall asleep all the time. So, audio. Sorry, data. Yeah. Errors are errors, right? So you might omit to enter something. But what are data entry errors? Well, hopefully are something that you need to build in some checks, right? In code, they're called assertions. Assertions means you do a little check to make sure something is true, like check this column and make sure all the values are between X and Y, right? You don't have to worry about that kind of thing when it's coming off Cell Ranger for your single cell data because nothing has gone through human hands. But if something has gone through human hands, like a list, right? A list is supposed to be A or B. If you are working with somebody who's doing data entry, you have to give them very specific specs for how to build the table so it's computable. Speaking of data entry errors, right? Because people might type it in 5 different ways and a human being can understand it, but then the computer can't understand it. So then, when I was working with this wet lab that generated a lot of data and was giving me the spreadsheet with all the metadata information, metadata is sort of information about the samples. It's not the data, it's about the samples, metadata. I would say things like, okay, for this column, the value should be A or B. That's it. Or you give them a template like an Excel spreadsheet, which only allows specific values in the column. Why? Because then the data are clean and minimizes errors. These are all kind of like, they make the workflow a lot more streamlined. And as somebody if you're getting data from somebody who's not used to analyzing it on a computer, you have to set the system in place so that, you minimize these sources of errors. Because then in the code, if you say, split the samples into cases and controls, but you spell cases with capital letters and controls with capital letters, and maybe for like a subset of them, it's not capital letters, it's something else, right? Then your code isn't going to pick up all the samples. These are all very simple things. These are not the fancy things, but it sort of builds, exactly where your data are at then.
Like dealing with data sets that were collected by other people that you're.
Yeah.
What if I'm just looking at how a population of cells in a class have responded to some a force that I'm applying, right? And
Right.
I'm just generating that data myself.
Yeah.
You still expect missingness in that situation as well, because you're generating and curating your own data, so you avoid for that.
No. So if you are preparing a tissue sample of some kind or something in a flask, and then you are generating some kind of, I don't know, single cell metagenomic data or something like that, yeah, you're not going to have missingness there. You might have a subset of samples for which there is no RNA-seq because you ran out of budget or something like that, but you're not going to have that kind of missingness. Then you have to do things like make sure that the genes, make sure that you don't have an unusual dropout in the number of genes because you under sequence a sample or something like that. Control for sequencing variation. There's a variety of situations in which you generate and analyze data. So it's hard. So you have to, these are just general principles, and you just have to sort of see which one fits in your situation.
How people like build up questionnaires, it's a very consistent example that's given up, but just wondering how this applies to, wet lab stuff.
Yeah. In some cases, it may not apply in the case of missingness, but you're still going to have biological and technical sources of variation. Just the nature of the variation is different. Right? Yeah. Yeah, and I think as a field, we're trying to find a way to streamline this process, identify it. You might even, I think that as a person who is in charge of running a project like that, you might want to come up with like a pipeline to identify all these sources and address them, and then maybe get like a summer student to come in and help you clean the data, right? Before you, so, and it's iterative, right? So you run what you can through, and then you fix the data and you run it through, and then you compare and contrast and stuff. So yeah. Okay. Yeah, and the other thing is some measures might not pass QC, right? So if you spend money and generate whole genome bisulfite for a bunch of samples, and some of the samples consistently fail bisulfite non-conversion or whatever the QC metrics, you are probably not going to put them in, or you might do analysis with them and without them. And if the results hold, then you just put a caveat in your paper. Like, so missingness can happen along the way. I'm sorry?
That kind of sounds like Thomas, kind of sounds like back from samples. So,
I think the more broadly, the point is that you need to be aware of these categories that can introduce bias in your analysis, right? Some parts of it might apply to your particular workflow more than others. But if you have a checklist, which is what I recommend, then you can just be like, okay, this does not apply to me, so just move on, right? But you have to know it. I can't tell you. Like, I've been at projects where you've got like multimodal data from patients, and the people analyzing it have not explored the missingness in the data. And then you go look at the missingness, and I'll show you some examples, right? And the way you then handle that is different. So this is just the purpose of this lecture, is to just introduce you to the variety of things that are going to happen and give you a checklist to systematically look at it. And then you decide what applies in your case, right? So what do you do if you have missingness, right? One thing you can do is you come up with a metric for what you consider excessive. Patient has less than half the data or whatever, you remove it. Try to stick within your field conventions, because at some point you're going to want to publish this, right? So make it defensible. Or you can use techniques such as imputation, which help guess at the missing values, okay? And again, it depends on how much you're imputing and whether it makes sense to impute. Again, the point is, do not just throw it into a pipeline. Just because there's a package that'll do it and give you some numbers doesn't mean those numbers make sense, right? Like, you can impute some things. Are you really going to try to impute, clinical variables? In some cases, maybe they make sense, maybe they don't. So just think more deeply about your data as a general point. But there is imputation techniques exist. We're not going to cover it here. Ensure your approach is defensible. So what are the trade-offs, right? You could be really conservative and say, anybody with missing data, I'm just going to take it out because I don't trust it. Now you've got like a fifth of what you had because there's so much missingness in your, So then you have to, what I would advise is you have 2 or 3 different scenarios. One is remove everything. One is remove the most egregious things. And then the third is like keep it and impute it and just run them all. Just set up your pipeline, because now you know how to program, and run them all and store them in separate data folders on your computer so they're not mixed with each other, right? And like date them so you know when you generated it, and then see what it looks like. So this is sort of the art of data analysis, right? There's like a programming is like push a button, ran a pipeline. But the art of data analysis is like you clean things, you compare the different scenarios, it's all looking consistent, or they're like wildly different, then you're in trouble, right? Okay, so that's trade-offs. So this is a matrix of black and white where the white indicates missing data and the black indicates existing data, right? So a visualization like this, and I think in module 3, we give you a little subroutine that does this, and it may not apply for things like your omic generated data sets, right? But this is one way to deal with missingness is how do you look at it, right? So you can look at it in black and white, and then you start to see patterns, right? So here, what is the pattern of missingness here? It's just one column, right? So it might be one patient or one sample that's got a lot of missing data. And then the question is, why did this happen is another thing, especially if you've got a bigger project, you want to fix the process, right? So look at your data, look at your data, look at your data. Here is an example of unstructured missingness. There's a few missing things, they're sprinkled all over. Maybe we can impute them. I don't know, try it, right? This is called structured missingness. Like it's just a loose term. It just means that, you've got greater than a certain amount of a column or a row or a chunk of your data set that's missing. This is an extreme example. This is biased structured missingness. All of the cases from one city did not have their proteomics done. So because whatever, we haven't done it yet. So if you don't look at it, you don't notice it, okay? So this is just know that missingness can come in different flavors, okay? And again, if you were to just take a matrix, you're in your R studio, you open the data table, and then you're just like, I'm just gonna impute. Hi, welcome.
Come on in.
Yeah, please sit wherever, take your time. You just coming from the airport? Oh my goodness. There's breakfast and coffee and
The Wi-Fi is on the board. It's the OICR guest network. Just click login. Let me know if you have any issues.
yeah, so just relax and then we can cover the stuff you've missed later. Yeah, everyone's like the coffees and the Wi-Fi, and then what else do you really need? Yeah, so just the point is that if you this is just to make the point that if you're just applying imputation blindly, right? And this point is gonna come up with clustering as well. It's like if you make decisions in your pipeline without having looked at your data, and your data has some very biased patterns in it, you missed it, right? You would have probably done things differently. So what if you had analyzed this and then you were like writing it, you present it at conferences or whatever. I am definitely trying to freak you guys out. You present it to your peers and then you come back and you're like, oh my God, I've been imputing things and this very critical subgroup, doesn't have this data or there's something weird about it. Find out this kind of thing sooner rather than later. So what are the goals of EDA, exploratory data analysis, right? The goals are to identify the magnitude of known biological and technical variation. So known biological technical variation, you have to brainstorm. What does that mean in your case, right? To identify sources of unknown variation. So you cluster your data and you're seeing 2 clusters, but everybody swears there's no batch difference, there's no lot difference, there's nothing. Somehow but the data are saying something different. So you can identify it, can quantify it, you can model it, you can regress it from your model, but you have to look for it first. You're going to ask something? You look like you're going to ask something. Then detect outlier samples. This is another big one, right? Is, why are they outliers? Maybe they got mislabeled. I did an EWA study on schizophrenia and bipolar disorder where we got postmortem brain samples from a bunch of different biobanks. And in some cases, it said male, but it clustered with the females. In some cases, they were duplicates because we did the SNP arrays on them and they were identical, right? And we did it on the neuronal fraction. So we took the brain samples and we sorted neuronal cells for our EWAS. And then we had some non-neuronal samples. And there was this one neuronal sample that was sitting with the non-neuronal, right? These things happen. We're human, right? But there are ways to pick it up, and you should characterize missingness. Okay, so we are going to articulate these goals so that EDA isn't just this amorphous thing. You've got a checklist of things to address, and there are techniques to do it. So how do you do this, right? These are the 4 goals as before. How do you know the magnitude of known biological and technical variation? You do things like principal components analysis for PCA. You do clustering, prior knowledge in the field. They know microarray lot numbers are an issue. So you color code your samples by lots and you see, is this a difference? Because at the end of the day, there could be 45 sources of variation. If you model them all, you're going to lose statistical power. So you need to be able to see which are the big ones to model and the ones that you have to have for sure, like genetic ancestry PCAs. No question, you have to have that in there. Otherwise, the reviewers, or if nothing else, the reviewers are going to come say they want it, right? But also, your inference could be wrong. But there are others that are really minor. So you might just kind of on the side do a smaller version of it. And you're like, oh, I still see the same thing. It's more weak, obviously, because I put more terms in my model. So, but at least you know that it doesn't go from A to B. Identify unknown sources of variation. Again, these techniques, dimensionality reduction and clustering, are going to tell you something. There are some packages out there, such as surrogate value analysis, which are going to quantify. They're going to say, give me all the variables that you think your model should have, and I will tell you if there are additional sources of variation, right? And it'll say, I found 2 surrogate variables, right? And just take these and build them into your model because your data have this structure. So there are methods to do that. And then detecting outlier samples, again, clustering, visualization, there are ways to quantify it, right? So you can say, find me samples that lie 2 standard deviations away from the mean, Z-score, whatever. Like there's ways to quantify it, right? Exclude samples from the analysis would be the solution. There's a big caution symbol here, right? Because, I was working with a biostatistician and he was like, oh, excluding samples, like a no, because it's a bias. You're introducing bias and it's a slippery slope. And when do you stop excluding? So this has to be done with extreme caution, right? Outliers. Maybe you'll do the analysis with it and without it. The same old deal, right? There's ways around it. Characterize missingness, and then you make your strategy. You plot it, and then you figure out your strategy. If a sample is missing 90% of the data, there's no point in having that sample in there. Maybe the field agrees that's reasonable, right? And then define your imputation strategy. So again, as in informatics, there are many tools to do the same thing. Which one fits your bill? So then you have to figure out, okay? So here's a little best practices checklist for exploratory data analysis. This assumes you've got, 2 tables for a data set. The one is measures, and it's a long, skinny table because you're doing 20,000 measures in, I don't know, 50 samples or something like that. Whoops. And the second is the sample information, the metadata, which has got the sample information. I don't know which flask it was grown in, which pond it was collected from, other variables that are of relevance, age, sex for human stuff. For mouse, it might be the age, whatever, things that are sources of variation. So then the other point I'm going to make is in R, learn how to explore your data in simple ways like using summary, the summary command. You guys learn about the summary command? Table command, right? What does a table command do? Category. Yeah. So if you need to print out a cheat sheet for yourself with these basic things, do it. Number of rows, number of columns. And I would say when you are building out something, try to do it interactively and look at these things. How many rows in my table? How many columns in my table? There's another one that's like sum is NA, right? Which counts how many rows or columns are NA. There's shorthand in R, things like row sums, which will just kind of add up the sums in a matrix. So you can say row sums is NA, right? And what that'll do is on each row, it'll tell you how many NAs are in your data set. So there are things like this. You just need to figure out what they are, make a little cheat sheet for yourself, get into the habit of just sitting and looking at your data rather than just throwing it into a pipeline because it's big and unwieldy. And the pipeline gives you a way to control it. Like embrace learning to look at your data, learn how to print things like how many rows, how many columns, okay? You guys know about the sprintf command? Yes, sprintf command? Okay. It's just a way of printing numbers into like a sentence, right? Sprintf, like you want to say, I have %I rows. I hope you guys kind of see this. I have %, I'm going to put that here. Sprintf, I have %I rows and %I columns, right? And then this thing is a placeholder for numbers. And then you can have like number of rows in my data frame, number of columns in my data frame. And if your R code has all these sprintf commands, it'll just print these things every time you run it, right? How many genes came up significant? I excluded poorly expressing genes. I went from 2000 genes to 5 genes. Oh, wait a minute, what happened? Why did it go down to 5 genes, right? So you do these little printouts. As printf, this is the command, you can go look it up, but it's a way of making sort of printing numbers in sentences and stringing them together rather than just doing print this number, that number, which is, it's inarticulate, right? It's like if you say print number of rows, your code will say 45, 62, 23.57896. And you're like, what is this? Sprintf makes it kind of literate. Okay, so initial exploration. How many measures in my metadata? How many measures in my data? You should know this. When you are working intimately with a data set, you should know the numbers like the back of your hand, so that if something goes off, you know it. Is there missingness? Decide a solution, right? For some cases, there might not be, like omic data generation. Examine the data correlation structure, which is basically what clustering is doing, right? All of the in single cell, I visualize it, all of the similar species hanging out in the same part of the PCA, right? That's correlation. That's one of the reasons they're like all together, or all of the glutamatergic neurons are in one side of the plot, right? And then the GABAergic ones are next to it. So examine data correlation structure. Does the data have natural groupings? How many major sources of variation are there? And PCA can quantify this. What do they correspond to? Right? Like something explains 80% of the variation in your data. Oh, it's batch effect. Better correct for that, right? Expand your model. Are there batch effects? Attempt removal or model it in. Are there unmodeled sources of variation? Surrogate variable analysis is something we use in bulk genomics. I don't know what people use for single cell. I've never used this single cell. And then you generate your final model for inferential testing. Inferential testing is where you say, which of these measures is different according to my variable of interest? Which of these genes is disease associated, treatment response associated, whatever associated. So that's your final model. Okay. And here we have some tools listed for each of the steps, right? So like learn to use all this stuff to explore your data, count NAs, imputation to interpolate, clustering to look at correlation structure, dimensionality reduction for source of variation, and you use very similar tools for all these other steps. And then finally, you want to use your models for inferential testing, linear models, generalized linear models, EdgeR, etc. for your, that's what it's doing under the hood. So if you have single cell data and you say Seurat find markers, at some point, that's what it's doing. It's fitting a model for each gene. Okay. So clustering. Okay, so the purpose of clustering, it's like, what did I intend to put here when I just set the word clustering at the top? The purpose is to find groups in your data, right? Nothing fancy. Your purpose might be finding batches in your data, finding patient subtypes, finding out how many populations you have, right? Finding groups of co-expressed genes. Maybe you are studying cell differentiation and your goal is to identify the program of genes over time, like gene regulatory networks. Right? So maybe that's your goal. But everything starts with clustering. So this is called a dendrogram, right? And it's basically, in this case, it's showing you the similarity between your samples. And this one has been clustered on the columns and on the rows. And this is, I think, the demo data we're going to be working with. And what it's done is it's grouped the samples based on their similarity, and it's grouped the genes based on shared expression patterns. So it's finding you sets of genes that are expressed together. What does it mean that those genes are expressed together? That depends on your application. Okay. So another application of clustering might be to find out how many entities you have. So in tumors and in cancer, there was a big wave of trying to understand how many types of X cancer do we have? How many types of colorectal? How many types of glioblastoma, which is a brain cancer, do you have? And you take your data layer, and you cluster it to say, oh, okay, I think we have these 4 subtypes of cancer. Another thing you might have is in single cell genomics. You identify, so what you're looking at here is single cell data from the developing human cerebellum. And each of these has been colored based on clusters identified by an algorithm, right? And then what do they correspond to? That the biologists have to infer by plotting the known marker genes for major cell populations and saying, oh, look at this. Cluster 1 is expressing Purkinje cell markers. So we're going to call this, and nothing else is expressing Purkinje cell markers. So we're going to call this the Purkinje cell cluster, and so forth. The heart of clustering, speaking mathematically, is finding the distance between samples. Okay, so you need a way to quantify given, 2 samples. Suppose you've done RNA-seq on them. So now you have 20,000 numbers for each of the 20,000 genes. Just keep it simple. Let's not talk about transcripts. 20,000 numbers per sample, right? And you've got 2 samples, A and B. So now you're looking at 20,000 dimensional data. okay? This is why we talk about dimensions in bioinformatics, like in genomics. It's because we do thousands or tens of thousands of measures per sample, per cell in single cell, right? And you know, at the end of the day, you have to figure out how to cluster these. So luckily for us, mathematics has developed the solution for this. We have formulae for computing distance between samples. So what's an example? Can anyone throw a distance metric at me? Anything from school, high school even? If you're given two data points that are on like an x-axis and y-axis, you want to compute the distance between them, right? We'll go through it, don't worry about it, right? But suffice it to say that you have a vector of numbers per sample, and there is a way to compute a distance between it. How similar or dissimilar are they, okay? And again, different data types need different distance metrics. This is why you have so many options for metrics. The kind of distance you compute for gene expression data is different from the distance you compute for sparse, sort of count-based data is different than the data for, sorry. So if you have continuously valued numbers, like from a microarray, you would use a different distance metric than if you have discrete values, right? It would be very different if you had clinical questionnaires with a score, right? Score everything from 0 to 5. And then you want to cluster people based on their responses. You have to pick a distance metric appropriate for that, which might not be the same as the distance metric you use for gene expression data. The point is just, you have to know what flavor of distance metric you have to use for your data. And again, the way to do it for any given data type is to find, landmark papers in the field and what pipelines they suggest, or come attend courses like CBW, where we will say, like, is this the kind of data you want to analyze? This is the workflow, use this distance metric. Okay, some data types have many distance metrics. Okay, fine. So here is sorry, you have a question? Yes, yeah. Metric just means how do you measure this, right? How do you measure similarity between two samples? What's an example of a similarity metric? Nope. How do you measure? If I tell you, if I give you two sets of, two RNA samples, and I say, I want you to tell me if these two are perfectly similar or not, or how similar those two are. You guys all know this. It's getting there, but it's even simpler. Correlation, right? You could correlate the two samples. If they're perfectly correlated, then, their distance is like 0. Pearson correlation, Spearman correlation, right? They're two faces of the same coin, similarity and distance. It's just that mathematically speaking, distance, you can build things out from with clustering, right? So you can say distance equals 1 minus similarity, but although that works in English, it doesn't work in sort of like whatever the matrix algebra is that's underlying this. So for example, here is Euclidean distance, right? Which is a very simple distance. So if you have two sets of numbers and you want to, like, gene expression for patient A and gene expression for patient B, and you want to compute the distance between them, you can just use an R function that computes Euclidean distance between them, right? So let me see if I can get a more high contrast because reds are usually not very high contrast. That's not very good either. Let's try the black. Okay, that might be better. So let's go through a worked example of Euclidean distance. That's it. This is the formula, right? If you have two measures per sample, suppose I only measured two genes per sample, A and B, right? Then I can plot Can I remove this here? Yeah. Where's the eraser? So this is kind of important to wrap our head around, so we're just gonna step into it a little bit. Okay. So, suppose I have two samples, A and B, and I have gene 1 and I have gene 2, okay? And I measure them in both of these people. So suppose gene 1 is at 10 units and gene 5 is at 5 units. Gene 2 is at 5 units. And suppose for B, gene 1 is at 2 units and gene 5 is at 5 units as well. Okay? It's just the numbers that came off the sequencer or whatever. All right, you're putting. So how do I compute the distance between A and B? Okay? So let's say this is A, gene 1. This is gene 2. Okay? Now, A is going to be at 10 for gene 1 and 5 for gene 2. So that's where this guy is going to live, right? He's at 10, 5. B is at 2, 5, right? Because what we found is that if we don't walk through this, students find like all distance and clustering, it gets really abstract. So if we understand it in 2 dimensions, then you can kind of understand it's the same thing that's happening. It doesn't matter how many measures you have, it's the same thing that's happening, right? So data point 1 is at 10, 5, and data point 2 is at 5, 5. So he's up here and up here, right? So 5, 5. Well, let's make it orange. So let's assume we know who this is. This is B and this is A. So what is the difference between B and A? There's a distance between B and A, right? This formula, we just plug in this formula, right? It's x2 minus x1, x2 minus x1. Can somebody read that for me?
X2 minus x1 squared plus y2 minus y1 squared squared.
Okay, so now x2 is the value for B here, right? So it's, you can see what's happening. It's B's x value minus A's x value squared plus B's y value minus A's y value, which is 5, right? Squared. And so this is minus 5 squared plus 0 squared square root. Okay. So this is square root of 25 plus 0, which is 5. So the distance between A and B is 5, right? So now, here we only have 2 dimensions, X and Y. If you had 3 dimensions, you measured 3 genes, then you would have a Z2 minus Z1 squared. If you had 4, you'd have a W2 minus W1, and so on and so forth. So now you understand when you compute a Euclidean distance, it's just plug in a formula, and you get a number out of it, distance between them, right? So if you have another guy, C here, which is like, maybe C is here, because he's the same cell type or whatever as B, his distance value, is it going to be smaller than AB, or is it going to be larger than AB? The distance BC. Is that going to be smaller than AB, or is it going to be larger than AB? Smaller, right? That's it. And everything else the computer is going to compute for us, right? We just have to know what is the metric. And there's other formulas for other types of distance, right? So why are we talking about distances? And why are we going into this level of math? It's just in part because things get very removed from this layer. This is sort of the heart of it. You know, once you figure out the distances between your samples, then you can use the clustering tools, because that's what the clustering tools are based on. You use these measures to evaluate the goodness of your clustering. They're all related. So it kind of helps to understand under the hood, like, what does it mean, distance between my samples, right? Numerically, that's the kind of thing it means. It's in green, so you might have to walk up close to it. We'll do it in black next time. But at the heart of it, and also this is the terminology is something which can sound really sci-fi, dimensionality reduction, but it just means that, you know, we measure 20,000 numbers per sample, but we can't see in 20,000, you know, dimensions, and not all of them might be relevant. So we're gonna boil the data down to the major source of variation so we can see it on a plot. So like, how is it that you see those single cell data sets on an XY plot, right? Somehow you are taking those 20,000 numbers you got per sample and you're boiling it down into like 2, right? That's not just picking the top 2 genes, it's some linear combination of it, right? So at the heart of it is just that you have to understand that when you make a omic measure, you are measuring a set of numbers, and then you want to find out sample similarity, etc. Under the hood, these are the tools, distances, right? It's not that hard, and you don't have to like code this from scratch. There's a dist function in R, right? And you can say what flavor of distance. So all of this is there, but as people who analyze your data, you have to kind of understand, why do we say it's important to pick the right tool for a particular data? Because, you know, maybe it's making assumptions about how that distance is computed that's not relevant for your data. So you have to kind of know a bit of this, okay? There are different flavors of distances, just know this. Some do assume that there's no correlation between all of your genes. Others are specially tailored to find correlation, you know, and take that into account for the distance. Just know that there are different kinds. And there are still others, such as block distance, Manhattan distance, might be better suited for categorical data, right? Remember that in numbers, broadly speaking, you've got 2 kinds. You've got continuous numbers and you've got discrete numbers, right? Sequencing, count-based data, discrete numbers. And then you've got continuous valued data like RNA, microarray data, it's continuous valued numbers. Sometimes you convert from one to the other after normalizing, but you should just know that these are the determinants of, in part, what metrics you choose. Okay, so we have sort of different kinds of distance metrics. Here's another one called Hamming distance, which is a way of looking at the number of mismatches for categorical data. There's just different metrics. Okay, common clustering approaches. What are some common clustering approaches? Oftentimes, if you're in genomics, you will see a hierarchical clustering, okay? This is one type of clustering approach. You have another one called k-means, and I think we're going to briefly go into what they are. But as with everything else, when you say distance clustering, dimensionality reduction, there are multiple options. There are multiple options for distance. There are multiple options for clustering. There are multiple options for dimensionality reductions. Which one should you use? Depends on your data type. Again, it comes back to knowing what are the underlying stats for that particular data type and then picking the tools. And it's good for you to kind of know it, even though you don't deeply think about it every day. And you should know enough to go look up the best practices for your data type. Okay? If you don't even have an appreciation of it, you can take any old data, throw it into any old package, and then compute it and garbage in, garbage out. You'll get numbers, but those numbers may be filled with bias because you didn't pick the right models, right? So you have to appreciate some of this. So at least you pick the right models. There are different kinds of clustering, like, you know, spectral clustering. We'll just mention this as well. So how does hierarchical clustering work, right? Hierarchical clustering is stepwise grouping, right? So what you do is you start with, suppose you have 6 samples. You find the samples that are the closest to each other, and then you connect them with each other. Now that becomes a meta group, right? It becomes like a group. Then you take the group and you connect it to the closest sample, the sample that it's closest to. So now you have drawn a connection between the AB group and C, and DE is still kind of sitting there. Then you find that F is closest to DE, so you connect DE and F together. And then finally, you put all of it under one sort of one umbrella. So what it does is, that's why it's called hierarchical, right? So it's sort of it's not taking a big sweeping view of everything and deciding what to connect to what. It starts out with the 2 closest ones, puts them together, then it puts the other together, right? The fancy word for this is agglomerative, right? It just means you're doing this kind of gradual accumulation to build it out, okay? This is appropriate for some types of data, not appropriate for other types of data. What about if you already know roughly how many clusters you should expect? I have 5 bacterial species, just cut my cells 5 ways, and we'll take it from there, right? This is where k-means clustering comes into play. If you kind of have an idea how many clusters you have. So in this case, you tell the computer, I want you to give me 5 clusters, right? And what it does is, I'm going to show you the animation for this. So here's what it does. It takes your data and then it says, you want 5 clusters? I am going to throw a data point in 5 spots on this sort of map as a starting point for my 5 clusters. I'm just going to roughly kind of throw it all around 5. And that's called a centroid. A centroid just basically means the average, right? So if you have 2 numbers, if you have 5 and 10, and you take the average, 5 plus 10 divided by 2, 7.5, right? If you want to do that in 2 dimensions, you take the average in the x, and then you take the average in the y, and that becomes the centroid for 2 dimensions. And it continues this way. So if you have 20,000 numbers for each of your 20,000 genes, it finds the average for each of those 20,000, and then that becomes like a point, the centroid, right? So centroid is just a fancy word for multidimensional average. Okay, that's like a center. And so what this algorithm does is it sets the centroid. It says, oh, you want 3 clusters? I'm going to throw 3 random dots. Then I'm going to bin your samples based on which centroid they're closest to. And then now I have computed new clusters based on my binning, and I'm going to compute the centroid for each of these 3. Then now I've got a new set of centroids. Now I'm going to rebin my samples, and I'm going to do this over and over again. And so the centroids start shifting until you find centroids that don't move much, right? So if you are dramatically wrong in your first set of centroids, over time, the centroids will shift and they will start to stabilize. And it's got some kind of numerical criterion for the stabilizing. You are recording, right, Nia?
Mhm.
Okay, good.
Don't worry.
Yeah, so that's how k-means clustering works. The computer you say, I want 5 clusters, right? So what is the disadvantage of k-means clustering? That's right. You have to know roughly how many clusters you're expecting, right? But it's a reasonable sort of choice in some circumstances, and you can try different clustering methods. Again, this comes down to the art of data analysis versus the science of data analysis. Like you're going to have best practices in your field, but there is a fair bit of leeway in how you're picking it. And also you have to remember the big picture, right? You have to make sort of defensible conclusions, but this data analysis might not be the only piece of evidence supporting your overall conclusion for your project. It's just one piece of it, right? So the data analysis just has to follow best practices enough to get you to your conclusion, and then you validate it using other sources of data. So you don't have to like really sweat it, unless that's the only data you are using in to, make your conclusions and publish your paper, right? Or whatever. Then if that is the only source of evidence, then yes, the reviewer is going to ask you to like do it a million different ways and test it. Why did you pick 5 clusters or whatever? But a lot of times it's kind of semi-reasonable, and you'll know from your data set, right? So spectral clustering is used when the patterns of grouping are not based on things like Euclidean distance. So for example, right here, if you have data that looks like this, right? And you do k-means clustering and you say, I want 2 clusters, maybe this is wrong. Maybe this is the clustering pattern I need for my data based on my application. This requires domain knowledge, right? So there may be things like social networks and disease transmission, and you want to find clusters in that, like things that are not your standard, you know, omics kind of applications where the shape of the data are different. So you just have to be careful. Again, if you don't visualize your data, you don't do PCA, you don't look at it, you're not going to know your data look like this, right? There are a lot of these common examples in like introductory statistics classes that say, that show you that you can apply a method and get an answer, but the answer is wrong because you haven't looked at your data. And if you looked at your data, you would know that for that structure, you've gotten the wrong answer. So just to say that there are distance measures that use other paradigms, such as social network similarity, right, in order to achieve different clustering goals. See, how do you know how many clusters your data have, right? So suppose you don't know a priori that you want 5 clusters or something like that. Maybe it's 3 clusters, maybe it's 4 clusters, maybe it's 5 clusters. How do you pick, right? So there are metrics in the field to quantify the tightness of your clusters. So I think we agree on this heuristic that when the dust settles, something in a cluster had better be, hey, Delaram, something in a cluster had better be closer to the members of its group than it is to members of a different group, right? If I am a neural sample, but I am closer to a non-neuronal sample, maybe I've been misclassified, right? That is the assumption. So there are a number of metrics here, the silhouette statistic, Dunn index, connectivity. And again, you don't have to program these by hand. There are packages, and we're going to go through an example which where you can quantify it. Okay. So you can decide on the number of clusters. You can do this with hclust. There is an R package called clvalid, and it'll compute these measures. For example, here's an example of a metric for cluster tightness, okay? So the silhouette index of a sample tells you how close that sample is to its nearest neighbor compared to samples in its own cluster. Right? Does that make sense? It's just intuitively, it makes sense. It's like if you have a data point, if on average it's closer to things within its cluster compared to things in other cluster, and that's true in general of your setup, then your clustering scheme is finding tight clusters, separated clusters. But if your cluster assignment, which is the term cluster assignment, is messy, then you're going to have things that are closer to things in other clusters. That means your clustering isn't good. You have to cluster it better, right? That's right. It's just quantifying the tightness of a cluster. Like intuitively, you'd probably do this, right? You'd say like, oh, I have a blue cluster and an orange cluster, and all the blue dots had better be closer to the other blue dots than they are to the orange dots, It's just quantifying this intuition, okay? And so this metric, the best cluster separation is a positive number, and the worst cluster separation is a negative number. So here's an example where you've got 4 clusters, 1, 2, 3, 4, and each row is showing the silhouette index of a sample. And so you can see that these 3 little chunks are positive, and this little chunk is negative, right? So what this is showing is silhouette statistic, and it's saying these 3 clusters seem to be tight, but there's this 4th cluster that seems to have negative values. So maybe we shouldn't be doing these 4 clusters. Maybe we should be clustering the data a different way. In practice, you test multiple clustering methods, and then you use these metrics to decide which is the best one. Should I use 4 clusters, 5 clusters? This is just an animation there. This is just showing you like a data point. It's showing in the silhouette index, what is the distance of me to everybody in my cluster compared to me and everybody in the neighboring cluster, okay? Does the intuition make sense? Is anybody not clear on what's happening?
You're like little circles around the cluster.
No, this is what you use to decide which clustering method worked the best. I tried 5 different ways of clustering my data, and silhouette index was the best for 3 clusters. That means that when I pick k equals 3, I get the tightest clusters. This is just quantifying how tight your clusters are. Okay. Similarly, you have another method. I'm not going to go into the exact formula. Just know that there are multiple metrics. They have pros and cons. So on balance, you want to pick the one that most of the methods agree on. Okay, so the done index is something similar, right? And higher the number, better your clustering. Okay. And then you have a third metric called connectivity, and it's just a different way of looking at similarity. And if you minimize, this one's different. This one, the lower the number, the better. Okay. It counts what fraction of your nearest neighbors are not in your same cluster. Why do we do this? It's because different metrics have different, have their own limitations. This isn't true in informatics in general, right? So it's like every method has got advantages and disadvantages. So for your clustering, again, the art of data analytics, you do a bunch of the clustering ways, you try a number of different clustering approaches, and you compute these measures for all of them. And you see on balance, the metrics, you have 3 metrics and you take a consensus. They seem to be saying that this cluster is the best, right? But then again, this is just math. Is it meaningful? You have to decide that. Does it make sense that I grouped my things into 3 clusters, right? Is it telling me something about the biology of my study? I had 5 bacterial species, but there are 3 clusters. Does that mean that 3 of those clusters are so similar, they might as well be 1 cluster? Or should I force it to do 5 clusters? Or did I undersample the other 2 samples? Right? So those are things that the domain expert has to decide. So clustering, to summarize, helps you find groups in your data. Okay, they could be cell populations, they could be patients, they could be batch effects. It's just groups. What the groups mean, you have to figure out. Okay, at the heart of clustering is a distance metric. You compute the distance, you represent the measures that you got from each sample as a point, a data point in high dimensional space, right? And you guys can all say high dimensional space now with a perfectly straight face. I once had a PI who laughed at me when I said that. I was like, yeah, high dimensional. He was like, high dimensional. Anyway, represents a data point in high dimensional space, uses distance. You've seen what distance is. Very simple. There's a formula, R, blah, blah, blah. There's like subroutines in R to do it. Multiple clustering methods exist. Pick one appropriate to your application. Measure the goodness of your clustering approach with a bunch of metrics, and we're going to show you a package that lets you do that. For single cell, I've seen the field kind of does different things. So like you can measure silhouette index, but I've also seen people, you know, when you have a big single cell data and there's like 40 clusters, they will plot the cells in each cluster one by one and see if they will use a combination of visual inspection and tools like Clustree. There's like a software package called Clustree that just tells you, as I increasingly fragment my group into more and more clusters, am I just seeing a sub-splitting, which would indicate that maybe you can just pick the high level, you know, levels, or am I seeing a complete reassignment? So for every application, there are different ones. Okay, so to recap, what we did this morning was we talked about how to think about your data and know your data through the process of exploratory data analysis before you do the thing everybody talks about, which is the differential whatever analysis, right? The goals of exploratory data analysis, or EDA, are to identify major sources of variation and covariation, identify outliers and missing data, either for the purposes of cleaning or modeling it into your, your model for gene expression, or for improving your process over time, right? So performing exploratory data analysis in a systematic way, like the little workflow diagram I showed earlier, will, introduce some kind of structure that will reduce the chance that you forgot something. And if you find something else that works better for your application, build that into a workflow like this. But it'll avoid the situation where you're just sitting there staring at the data and you don't know where to start, right? So you can do this, how many measures there are, blah, blah, blah. Each of these can be its own little R function, or you can have a single R function that systematically does these things, right? And does the clustering and the PCA for you and all that. Finally, we talked about clustering. It can be used to find natural groupings in your data. At the heart of clustering, if you want to group things, you have to quantify how dissimilar they are using a distance metric, okay? And then how do you know how many clusters are in your data? There are metrics that determine how good your clustering is, how tight things are. A tight set of clustering will mean that things in a cluster are closer to that cluster than things in other clusters. And there's a bunch of metrics for it. There are packages for it, okay? So now we are going to look at how to achieve exploratory data analysis and clustering using R.